come to the next class. So under fractionation of protein, so proteomics is the analysis of species, is the analysis of a species protein complement that is expressed by its genome. But the complexity of the proteome raises significant issues for a researcher. There are an estimated 20,000 to 25,000 human protein coding genes that could code for less than 1 million proteins, especially when protein modifications are considered. These include N and C truncations, alternative spliced forms, and a large number of CO and post-translational modifications, including phosphorylation, glycosylation, deamination, sulfation, oligomerization, and N-terminal acetylation. Yes, so what we generally mean by fractionation of protein, plasma proteins are so many. We mentioned some. We mentioned the albumin, we mentioned the globulin, we mentioned the fibrinogen, yes, and some other ones. So when a test is required to know if one has a different kind of health condition, like multiple myeloma and the rest. So these proteins has to be fractionated first. What do you mean by fractionation? Have to be brought into different components. So fractionation of proteins of tissues and cells can take many formats that all inevitable simplify protein characterization, including how different methods of characterizing these proteins. So separating them into their different components. Albumin, we know that okay, albumin is the percentage, globulin is the percentage, fibrinogen is the percentage. Then during the separation of these proteins also, so that during the separation of these proteins, we know okay if the person has more of albumins, more of globulins, more of fibrinogen, more of this or that. It purifies the proteins and separate them into their different components. So we have different methods of separating these proteins. We have the sequential fractionation or the differential fractionation. Sequential fractionation is ideal for the identification of unknown proteins, as no specific knowledge of the protein is required to fractionate it. Now, there's no requirement for specific proteins or capture media, as the general procedure uses for differences in protein solubility. Some proteins are more soluble than the others. Now, the fractionation of a protein on its solubility can be performed by ionic precipitation. Okay. They also have the subcellular fractionation. Proteins within a cell are often localized to specific cellular compartment. There are some that stay more in the nucleus, some that stays more in the plasma membrane, and other areas. So during the process of fractionation of protein using this subcellular fractionation, it separates them into their different components, that's where they are located. Then we have the abundant protein remover, a relatively simple method for simplifying the proteome for analysis is to remove the highly abundant protein. So for example, you know that albumin is small, so remove the highly abundant proteins such as albumin and immunoglobulins. There are numerous techniques available for removing this protein, but most use some form of affinity remover. Affinity, one that is more, that can attach to something more than the other. Yes, affinity method of removing. We have the chromatographic technique for fractionation of a specific protein or a specific type of protein. Glycoproteins, for example, people can use particular characteristics to pull these proteins for the proteomic pool. Chromatography can be used to fractionate proteins based on their molecular weight. Some are heavier than others, their protein charge, some are anions or cations, their hydrophobicity, if they love water or not, then their antigen-ligand interaction. Numerous chromatographic techniques exist to fractionate protein pools, and often more than one is required. However, all of them depend on having knowledge of what? Your protein, and all lead to a loss in protein. I think I've said so many things. Concerning protein, its fractionation, paraprotein.